Robbins and Cochrane. Pathologic Basis of Disease. Chapter 1: The Cell as a Unit of Health and Disease. Pathology literally translates to the study of suffering. The Greek pathos, suffering, logos, studio, more prosapally. The term pathology is invoked to represent the study of disease. Germane to this opening chapter, Virchow coined the term cellular pathology to emphasize the basic tenet that all diseases originate at the cellular level. Thus, modern pathology is basically the study of cellular abnormalities. Therefore, diseases and the underlying mechanisms are best understood in the context of normal cellular structure and function. Base pairs. Within the genome, there are about 20,000 protein-encoding genes, comprising only about 1.5 of the genome. These proteins variously function as enzymes, structural components, and signaling molecules, and are used to assemble and maintain all of the cells in the body. Although 20,000 is an underestimation of the number of proteins encoded in the human genome, given that many genes produce multiple RNA transcripts encoding different protein isoforms, it is nevertheless startling to realize that worms composed of fewer than 1,000 cells, with genomes of only about 0.1 billion DNA base pairs, are also assembled using about 20,000 genes to produce proteins. Even more surprising is that many of these proteins are recognizable homologs of molecules expressed in humans. What then separates humans from worms? The answer is not completely known, but the weight of current evidence suggests that much of the difference. The genome. The sequencing of the human genome represented a landmark achievement of biomedical science. Published in draft form in 2001 and more completely detailed in 2003, the information has already led to remarkable advances in science and medicine. Since then, there has been an exponential decrease in the cost of sequencing and an exponential increase in data accrual. This new information, now literally at our fingertips, promises to revolutionize our understanding of health and disease. However, sheer volume of the data is formidable, and there is a dawning realization that we have only begun to scratch the surface of its complexity. Uncovering the relevance to disease and then developing new therapies remain challenges that both excite and inspire scientists and the lay public alike. Plasma membranoprotection and nutrient acquisition. Plasma membranes and all other organellar membranes are more than just static lipid sheaths. Rather, they are fluid, the layers of epitophic phospholipids with hydrophilic head groups that face the aqueous environment and hydrophobic lipid tails that interact with each other to form a barrier to passive diffusion of large or charged molecules. A Fig. 160. The bilayer is composed of a heterogeneous collection of different phospholipids, which are distributed asymmetrically, or for example, certain membrane lipids preferentially associate with extracellular or cytosolic phases. Proper organization of phospholipids is important for cell health, as specific phospholipids interact with particular membrane proteins, influencing their distribution and function. In addition, asymmetric partitioning of phospholipids is important in several other cellular processes as follows. Phosphatidylcysteine on the inner membrane leaflet can be phosphorylated, serving as an electrostatic scaffold for intracellular proteins. Alternatively, polyphosphatidylcysteines can be hydrolyzed by phospho-PC to generate intracellular second signals like diacylglycerol and inositol trisphosphate. Phosphatidylcysteine is normally restricted to the inner base, where it confers a negative charge involved in electrostatic protein interactions. However, when it flips to the extracellular face, which happens in cells undergoing apoptosis, a programmed cell death, it becomes an eat-me signal for phagocytes. In the special case of platelets, it serves as a cofactor in the clotting of blood. Glycolipids and sphingomyelin are preferentially expressed on the extracellular face of glycolipids, and particularly ganglioside, with complex sugar linkages and terminal sialic acids that confer negative charges, are important in cell-cell and cell matrix interactions. Brain is liberally studded with a variety of proteins and glycotropins involved in 1-ion and metabolite transport, a 2-fluidophase and receptor-mediated uptake of macromolecules, and a 3-cell ligand, cell matrix, and cell-cell interactions. Proteins associate with the lipid bilayer by one of 4 general arrangements. How they integrate into the membrane informs function. Most proteins are integral or transmembrane proteins, having one or more relatively hydrophobic helical segments that traverse the lipid bilayer. Integral membrane proteins typically contain positively charged amino acids in their cytoplasmic domains, which anchor the proteins to the negatively charged head groups of membrane phospholipids. Proteins may be synthesized in the cytosol post-translationally attached to prenyl groups, e.g. farnesylate, related to cholesterol or fatty acids, e.g. palmitic or myristic acid, that insert into the cytosolic side of the plasma membrane. Insertion into the membrane may occur through glycosylphosphation, the GIA anchors on the extracellular face of the membrane. Peripheral membrane proteins may non-covalently associate with true transmembrane protein. Many plasma membrane proteins function together as large complexes. Of these may either be aggregated under the control of chaperone molecules in the rare or by lateral diffusion in the plasma membrane followed by complex formation in situ. The latter mechanism is characteristic of many protein receptors, e.g. cytokine receptors that dimerize or trimerize in the presence of ligand to form functional signaling units.